everyone, welcome to the course on medical biomaterials. We will continue on the topic of animal studies. The most important thing in animal studies is the animal ethics. Yes, especially when you are doing experiments with animals, we need to look at the ethical issues and we need to follow those ethical guidelines. Generally, any research organization, if they have an animal house, they will always have a animal ethical committee. So, this committee will look at what type of experiments that are going to be performed and whether they are abiding by the guidelines and so on actually. Okay. So, they have to get permission from the animal ethical committee before experiments are carried out in with animals. So, what are these? Animals are allowed for development of drugs and vaccines, and animals are used before they always go into human volunteer trials. Okay. And even when the clinicians or surgeons are looking at new surgical techniques, trying to understand certain new concepts, then they have to perform on animals. These animals, like I said in the previous class, it could be small animals, large animals, right, sometimes going right up to horse, starting from mouse. Okay. So, depending upon the type of animal studies they want to do. So, quite a lot of animals are used globally for research per year, and many countries have the mandate that animal studies have to be done before they are taken into human volunteers. There are three important guiding principles when you are looking at animals. They are called the 3 R's, okay, in animal research. One is called the replacement, other is called the reduction, other is called the refinement. What is this replacement? You need to understand whether there are any alternates available, okay, which uses non-animal studies. For example, can I use cell lines, can I use computer simulation techniques and so on. Of course, computer simulation techniques have not developed so much that it can completely replace animal studies, and similarly cell lines are not completely replica of animals. So, we need to still do animal studies. Reduction, can we use less number of animals, but at the same time get statistically significant information. Okay. So, can I use smaller set of animals? Can I do an implant on 2 sides of the same animal, that way number of animals can be reduced. So, you need to think about all those, that is called the reduction. Refinement, can I improve the experimental strategy so that the experimental strategy is well refined, so that the animals do not undergo too much pain, they have minimal pain, okay. The experimental duration could be short and quick, and the data is collected in a very efficient manner, that is called refinement. So, the main guiding principle in animal testing is the replacement, reduction and refinement. Okay. So, any ethical committee will ask these questions: can you reduce the number of animals, can you have experiments which does not use animals, or are you refining your experimental strategy so that the animals do not face too much pain, or the experimental duration is much smaller, and so on actually. So, there are a lot of guidelines, regulations so that the animals are treated humanely as possible. Okay. That is very, very important. Something called Guide for Care and Use of Laboratory Animals. There are certain guidelines available. The Guide for Care and Use of Agricultural Animals in Agricultural Research and Teaching, okay, like a sheep, goat, cow and so on. Report for the AVMA panel on euthanasia, in case animals have to be sacrificed, how humanly it is possible without causing pain. Guidelines for the use of fish in research, okay. American guideline which looks at animal care and use policies, Animal Welfare Act regulations, Public Health Service policies. So, there are a lot of guidelines available both in Europe as well as in USA for treating of animals of various types, for the euthanasia and so on actually. Okay, there is something called a committee for the purpose of control and supervision of experiments on animals. This committee I am sure has to be there in any organization which handles animal, which conducts tests of various types, be it drug discovery, where be it biomaterial studies and so on actually. So, this is the CPCSEA. This is a committee that have been empowered by law to ensure all research activities involving animals satisfies the federal, state and local regulations and policy, both governing the use of animals in research. Okay. So, that they have the mandate to look at all the research activities that are going on with related to animals. Okay. So, there is a chairperson, there has to be a veterinarian, a doctor who is an expert in handling of animals, scientist, okay, scientist who has done research in animals. Then there has to be non-scientific members. Okay. So, the non-scientific members looks at it in a different angle and tells whether the experiments that are going to be conducted is really important, whether the animals face pain and whether it is done humanely and so on. That is the non-scientific members. Then there are non-affiliated members, that means people who are not affiliated with the research organization, outsiders who are more interested in the community as a general, okay, community at large. So, people who are looking at it as a community based issues. So, those are also members who are inducted into this particular committee. So, we have a chairman, we have a veterinary surgeon, we have scientists who is going to be conducting the experiments, who is very well experienced. Then there are non-scientific members who does not have much science background, but they may have other background, and then members who are inducted from outside the organization. So, they are more interested in the welfare of the community at large. So, all these people form this committee and they go through the protocols or the experimental strategy which is designed, and they try to understand it, and they use all the 3 important principles, the 3 R's, and then make suggestions which are implemented before the experiments are started. So, what are the responsibilities of the CPCSEA? They review and approve all research, whether it is research, teaching or testing activities that makes use of animals, okay, small animals, large animals and so on actually. They look at whether there are no alternatives to using animals and the research is not duplicated. If the data is already available, why carry out that research? So, they might not approve if it is a duplication. And they also look at whether these experiments are relevant to human or animal health. Okay. So, it has to have some purpose, especially maybe if it is a drug for human or it is a drug for animals. So, you need to test it out a priori before they are actually tested on human or animals. Okay. So, whether it is relevant and it is good for the society. So, it is not that I want to do an experiment just for the sake of doing experiment, but there has to be an end point where the society at large comes into picture. So, we need to consider that angle as well. Okay. So, the CPCSEA looks at all these aspects before they actually give permission to the organization for conducting the experiment. So, they look at the procedures. So, they look at each and every protocol and then approve it to be conducted on animals, and then they identify potential painful and stressful procedures. Okay. So, if there are going to be any procedures that are going to be painful, can the animals be given medications, okay, or can we completely eliminate these procedures, or can we minimize this pain and distress. And then they also monitor the research activity to ensure only whatever has been reviewed and approved by the CPCA is followed. Okay. So, it is not that the experiment does something different from what has been approved by the CPCA. They also inspect the living quarters of animals. Okay, maybe twice a year they come and see whether the animals have a good facility. Animals are checked daily, their housing facilities are clean, and the space for each animal is sufficient for their well-being as well as for the production, and that they are regularly received fresh food and water. They are appropriate for the species being used. For example, if it is rats, there are certain guidelines in which they have to be housed. If it is a guinea pig, there are certain guidelines. If it is a bigger animal like dog or sheep or goat, then there are certain guidelines for their housing, for their food and water. So, whether they are followed and they are receiving proper veterinary care, that is also important. So, there has to be a full time veterinary surgeon who takes care of the well being of these animals. So, that is the overview of the CPC-SEA. Then of course, there is another committee that is called the Institutional Animal Care and Use Committee, okay, IACUC. So, they look at whether the number of animals are enough to yield the statistically valid results. So, they neither want to have too many animals nor too little animals which are not statistically significant, whether the appropriate species of animal is being used for the project. Like I showed you some time back, if you are talking about cardiovascular stent, what type of animals, if you are looking at joint, what type of animals, if you are looking at just cytotoxicity, what type of animals, and so on. So, the appropriate species of animal. And humane experimental endpoints have been established. That means there has to be a start and end. We are studying the biodegradability of a polymer over a period of 30 days. So, that is the start and end point. So, that is also very important. So, there has to be an end point. So, it is not an open ended research which they are doing. And then whether they are using appropriate methods for euthanasia, if the animal is sacrificed, okay, how it is being done, what procedures are being done so that the animal does not face pain when it is sacrificed. So, all these issues needs to be looked at, and which is being looked at by the Institutional Animal Care and Use Committee. So, there are quite a lot of overseeing committees which not only monitor the well being of the animal, but they also review the experimental procedure strategy. They give a lot of suggestions, modifications with respect to the 3R, and if there is any animal has to be sacrificed, how painlessly they are to be handled, and so on. So, these committees take care of all these issues. Okay. Okay. So, let us look at a few case studies which we have performed using animals, looking at biomaterial with respect to their biocompatibility or biodegradability. Okay. So, imagine I am looking at the biocompatibility of a certain polymeric system in vivo. I might have a priori done with cell lines. I talked about an assay called MTT or MTS assay, which looks at cell proliferation. So, after doing that, you want to move to the animal and look at the biocompatibility in biological environment, because when you keep it inside the animal, it is facing enzymes, proteins, so on. So, the way these material behave there may be very much different the way they behave in cell line. Okay. So, we had done some experiments. I want to show you some of these results. Okay. So, we were looking at in vivo biocompatibility and biodegradation of a polymer called polylactic glycolic acid, which is supposed to be an alternate use in stents, urethral stents. Currently polyurethane is used. So, we want to look at this PLGA in animal model, male Wistar rats. So, we have about 6 rats per batch, so with the control and with the test and so on. So, the age, weight about 6 weeks, 180 to 200 grams. So, these polymers are placed intraperitoneally and then sewed back, and after 30 days they are removed, and then the biodegradation of the polymer can be studied using weight loss, and by looking at the tissues one can look at whether the polymer causes any cytotoxicity or whether it is harmless to the surrounding tissues, and also look at systemic toxicity, whether the polymer in those 30 days causes toxicity to the system as well. So, this type of cytotoxic studies, these are very normally done. You can do it for short period or medium period or long period. Like I said, around 30 days is what is studied if you are talking about longer duration studies. Okay. So, after 30 days you take out the sample out, which is placed intraperitoneally, and look at not only the changes in the polymer you can also look at any changes, systemic toxicity to the rats as well. Okay. Then you can take out certain tissue samples, okay, tissue samples from the heart, and then see whether there is changes after it has been placed the polymer for 30 days. So, we can prove that there are no changes in the arrangement of myocardial fibers. We can take out kidney samples, and then we can see whether there is any changes with respect to control. We can look at the liver samples, and then with the control as well as with the polymer placed, and see whether any changes has happened, and also we can take tissues from the brain and see there are no inflammation. So, this type of studies are commonly done to look at the cytotoxicity, both the acute as well as chronic, and also systemic toxicity. So, we can take tissues from different parts of the animal and see whether there is a change because the material has been placed for 30 days intraperitoneal with respect to the control. Okay. Okay. So, similarly, okay, that is with respect to inflammation. Then we can also look at whether there are any giant cells that have been formed, whether there are any connectivity tissues have been formed, whether there is any collagen network that has been formed, and so on. Again, here I am showing you an example where we have looked at polyurethane. Polyurethane is widely used in stents, they are used in breast implants, they are used in many places actually, in tubes and so on actually. We have two different modifications that are done to polyurethane, and they are placed intraperitoneal for 30 days, and then we retrieve the implant, look at the histopathological studies. We can also look at fibrous encapsulation around that polymer. Is there any collagen that has been formed? In the previous study we talked about inflammation. Here we are talking about what are the changes that are happening to the connective tissues, okay, tissue response. Okay. So, we can sort of from the histopathology data we can count the number of tissue, different types of tissues that are formed, okay, and then we can sort of quantify them here. This is the control, these are two different modifications to the control. Okay. After 1 month of implantation we are looking at different types of tissues that are formed: acute inflammatory response, the macrophages, giant cells, okay, and fibroblastic tissue, collagen network. Okay. And then we look at each one of these polymer, effect of each one of these polymer. So, we say when we give a scale of 0 we say it is not present, when you give just plus or minus we say it is occasionally present, when it is present then we say it is present to a mild degree, when we say 2 plus then it is present to a moderate degree, 3 plus we will say marked degree, 4 plus very high. Okay. So, this is how it is scored. So, basically it is a qualitative representation of the observations. Okay. So, this is the control, we say acute inflammatory cells, it is found to a very high degree. Okay. Whereas in the modified system it is found to a marked degree, in one modification it is found to a moderate degree. So, there is a decrease in the inflammatory cells, okay, as we modify the control polymer. Macrophages, it is found in marked degree on the control, and then it goes down to almost a very mild degree. That means amount of macrophages found on the animal model on this modified system goes down from a marked degree to very mild degree. If you look at the giant cells, it is found quite a lot on the control, but it comes down quite drastically, which is say occasionally found. Same thing on fibroblastic tissues, from a marked degree it comes down to almost mild, and collagen network it is found to a moderate degree to almost to occasionally present. So, effect of these modifications with respect to the control is sort of determined in a very qualitative way, and it gives you a very nice picture, okay, on the formation of various types of cells network and inflammatory response. So, this is a very interesting study one can do using animal models, and as you can see we cannot do that sort of study in a in vitro cell line. We can look at the inflammatory markers like TNF alpha or IL beta, which I showed you, and because of the contact of the cells with biomaterial. But if you want to look at fibroblast, collagen network, giant cells, then obviously we need to do some type of animal studies. We cannot do it in the in vitro condition. And we can do some qualitative quantification. And as you can see in this table, a modification to the polymer brings down these tissue response dramatically with respect to the control. So, tissue response we cannot study in cell lines, but you have to study of course in a vivo model like a Wistar rat. Another example I want to show you, we were looking at wound healing, develop polymeric material for wound healing applications. So, we have create wound on the surface of rats, and then we look at how the various polymeric combinations, blends. This is a polymeric combination gel 25 using a glucon and a carrageenan, where 25% of it is a cyclic glucon, remaining carrageenan. Here same polymer, but with the ciprofloxacin, which is an antibacterial. When you have an open wound, bacterial infection could be a problem. So, we can see whether the ciprofloxacin helps in preventing the bacterial growth, and then this is a commercial product as a positive control. So, we have a control where we do not give any treatment, then we have a combination of cyclic glucon and carrageenan hydrogel treated wounds, and this is same thing with the ciprofloxacin, and this is a commercial product. So, here we are talking about 6 rats in each case. So, here we create an wound on the surface, and then these different treatments are given. Then you monitor how the wound area contracts as a function of time. You do these experiments for about 7 days and then see how it contracts as a function of time, and then we see whether there is a statistical improvement because of these type of new polymeric designs. And that is what one does actually. So, these pictures show you the wounds on control without treatment with the carrageenan and cyclic glucon. This is with the carrageenan, cyclic glucon and ciprofloxacin. This is a commercial product, and so on with different time periods, day 1, day 4, day 7 and so on. We can quantify the wound contraction area and represent it beautifully in this type of bar graph. So, what does it tell? As we go up in days, this is with the ciprofloxacin hydrogel, this is without ciprofloxacin, this is a commercial product, this is the control. So, we can see wound contraction is very high, very fast. Within 2 days we find a very high wound contraction when compared to the control or with the commercial product, and then the contraction rate increases dramatically within the 8th day when compared to say control. The wound contraction after 8 days in control is only 60% or 70%, whereas it is almost 100%. The wound is completely healed, as you can see here, in day 8, this is the day 0, and wound is still not healed fully in the control where you do not give any treatment. So, you can see this is with ciprofloxacin and with ciprofloxacin, this is a commercial product. So, this particular hydrogel seem to work reasonably well, and these type of experiments are carried out and have to be carried out using this type of animal models. Without that one could not go further for testing it out on human. So, we also can look at in addition to the wound contraction, we can look at whether the collagen network formation is good, so that the wound gets completely healed, because that is more of a tissue response. Okay. Like I have been telling for a long time, so you will have an inflammatory response, then you have a tissue response and so on. So, the major constituent of collagen is called hydroxyproline. So, we can monitor taking out samples from here, what is the hydroxyproline content, and which is a marker, biomarker it is said, which tells you how the wound gets contraction rate is good. So, as you can see here, okay, the hydroxyproline content is quite good, okay, when compared to the control or with ciprofloxacin and without ciprofloxacin. Okay, we see quite a big increase in the hydroxyproline content with respect to control. That means the network of collagen is also growing. One is the wound contraction, and then the collagen network also growing very well, so that the wound gets completely cured. Okay. So, the hydrogel treated wounds are moist, no sign of infection, inflammation, and the healing rate is also very high, almost more than 90% in 8 days when compared to the control or commercial products, and the high level of hydroxyproline, which gives you an idea about the increase in the collagen buildup, and the coverage of the wound is also very high with respect to the control. So, I have given you few case studies or examples where use of small animals is extremely important before one goes into human volunteers. These studies which I showed you with small animals, mostly looking at cytotoxicity, we can look at biodegradation of polymer, we can look at the wound healing properties, we can look at the tissue formation, and whether there are any collagen network formation when a material is implanted intraperitoneally. Of course, you need bigger animals if you want to look at, say, joint replacement, or if you want to look at cardiovascular stenting, because the rats have very small sized joints as well as veins. So, you may have to go for higher animals like dogs and sheep and so on if you want to look at other type of studies. Okay. So, the rats, Mr. rats or rabbits, okay, they are very extremely good for the type of studies examples which I showed you, just like such as cytotoxicity, such as biodegradability, such as formation of collagen network, such as inflammatory responses, wound healing, and so on actually. Okay. So, thank you very much for your time.